Pratnya?
Yes, Vidula.
Yeah. So I think we'll start.
Yeah.
We have still 5-10 minutes. People will log in by the time. I'll just give a quick introduction to Venture Center, TechX, and the speaker today. And then I'll hand it over to Dr. Ashutosh. So welcome to all the participants who have joined us online, and we also have a few participants who are there here in person. And Dr. Ashutosh Shastri has also joined us here. So welcome to all of you all for this event today on converting research into products. We will begin with a short introduction to what is Venture Center, then TechX, which is a vertical of Venture Center. And it will be followed by a very quick introduction to our speaker today. Then I will hand the mic over to Dr. Shastri. So Venture Center, this is an inventive business incubator, and we are a Section 8 not-for-profit incorporated as Entrepreneurship Development Center in Pune. And we started operations in January 10, 2007. We are the first incubator in the CSIR family, and we have been approved by the NSD, DVD, and BIRAC. We also have been recognized by DSIR and DIPP. And we have a CREDAL accredited NPO. This is basically more for CSIR eligibility. So all the corporate funding we can receive under the CREDAL accredited NPO. Venture Center has been awarded with many prestigious awards. A few of them have been listed here. First is the National Award for Technology Business Incubator, which was there in 2015. We have also been recognized by the Asian Incubator of the Year Award in 2018 and the National Entrepreneurship Award in the year 2019. The focus of Venture Center is basically to nucleate and nurture technology and knowledge-based enterprises for India by leveraging the scientific and engineering competencies of the regions in this region. Our focus area is promoting inventive enterprises and spin-offs from R&D organizations. We are sector agnostics, but our strengths lie in all science-based technologies. So few of the sectors we have mentioned it below. Now, the organizers of today's event, we are TechX.in. This is one of the verticals of Venture Center, and our focus is on tech transfer. So TechX.in, this is a tech transfer hub which has been established at Venture Center by BIRAC, and we have been supported by National Biopharma Mission. This is an initiative by Government of India DBT office, and our main focus is on technology commercialization. We help facilitate exchange and transactions related to any science and engineering based technological ideas. So to help all our partners and individual inventors with tech transfer, we have come out with different services. To name a few: technology commercialization, policies and IP protection and decision support, and industry academia partnering. So more details of what TechX does, you can visit www.techx.in website. Now coming over to today's talk on converting research into products, insights from my multidisciplinary problem-solving journey. To deliver this talk, today we have with us Dr. Ashutosh Shastri. Dr. Ashutosh Shastri is founding CTO of Immunotics, and he has a passion and expertise in building multidisciplinary teams, technologies and products for diagnostics and drug delivery. Utilizing his deep experience with microsystems engineering, Dr. Shastri led the product and technology development for Immunotics point of care system. Before joining Immunotics, Dr. Shastri served as Vice President of R&D at INDX Lifecare and as the Director of Engineering at Corium International. At Corium, he led the technology development and scalable pilot manufacturing of dissolvable microneedles patch from early concepts to phase 2A. Before that, he had led the development of a next generation infusion pump, which received an Edison Award in the category of science and medical device game changers. At the University of Washington, Dr. Shastri initiated research on super hydroponic surfaces for droplet microfluids. Dr. Shastri has a PhD in medical engineering and MTech and BTech degrees in chemical engineering from Indian Institute of Technology, Bombay. He initiated India's first BioMEMS project collaborating with Cancer Research Institute. He has authored many publications and also has 15 patents to his credit. He has served on Industrial Advisory Board for ULCA's Bioengineering Department, a National Institute of Health Review Panelist for Allergy and Infectious Disease, and National Science Foundation SBRI Review Panel for Medical Technologies. As a president of Epic Global, he reimagined it as a place where technology meets health and currently serves on its board of directors. So with this, I would like to invite Dr. Shastri to kindly take the talk further. I will stop sharing my screen.
All right. Thank you. Thanks for the opportunity to be here, and I'm happy to share some stories with you. So yeah, converting research into products. So, when I was on the other side of the aisle in academia, I felt the intellectual high point is the invention. And somehow that's very normal that academicians think like that somehow taking it from the concept to product is trivialized if you are in a university. But what I've learned to appreciate since is it's intellectually no less challenging to take it from that early concept to a reliably working product. So that's the story I'll share for one of the products. I'll tell you about two other products, and then give you a glimpse of my brush with inventions. So that's that's what we'll do today. So a lot of innovation is really taking tools from one discipline and applying it to another. So when you learn, so that's what at least my learning has been. So when I think of innovation, I think of synthesis. And in my case, the synthesis has been chemical engineering, electrical engineering, mechanical engineering, essentially applied physics and chemistry. So that's one. And second is anything worth doing requires a great team. So building and mentoring those teams has been as much fun and as crucial as developing the products themselves. And finally, taking these early concepts all the way to clinic is really about navigating risk. You want to carry minimal residual risk. So you're always prioritizing and picking your battles, and you want to take on the highest risks first. So that's what I'll talk through. Okay. So just a synopsis of what I do and love doing. In small companies, it's both fast-paced and frugal, and there are only so many people. So while you're leading the team, a big part is being a subject matter expert in a few things. So for me, that means microfluidics, both the stream microfluidics kind and the droplet microfluidics kind, and we'll talk a little of both of them. That's one of the areas. The other areas is biosensors, both optical, electrochemical, impedance-based, and microfabrication. And when I say microfabrication, it's silicon and non-silicon microfabrication. And thirdly, I think the surface area by volume ratio increases so much in microsystems that it's the surface forces that start dominating. So understanding the physics and chemistry of surfaces becomes crucial to solving problems. So that's the third area of expertise, so to say. And finally, in nature, everything works seamlessly. All these names that we coin, electrical engineering and chemical engineering and physics, are just for our own sanity and ability to cognize everything. But really, it all needs to work seamlessly as one system. So architecting those systems and integrating them so that they work like one whole is something I've learned to do over years. Yeah, so I think I'll skip on we already talked about risk assessment. So much of project leadership I've learned is saying no. See, logically, you can be doing so many things, but you have time and money to do only so much. So picking those things, okay, saying yes to a few and saying no to most is crucial to making startups meet their goals. And having that good insight and making good choices is what makes or breaks many companies, I felt. So yeah, and prioritization, I keep saying that if everything is priority one, then nothing is. I mean, it's just repeating a cliche, nothing special here. And finally, you have to have the sequence. You start with a product, the what to make, then you graduate to how to make, and then you start automating, which is what the pilot manufacturing is. Rushing to pilot manufacturing is automating before you know what and how is usually not a winning strategy. So anyway, I think I think just one thing I would, innovation by synthesis, I do want to emphasize this, that there is something called too deep because you have only so much time and money, but you need to be deep enough to be consequential. So that's again a judgment call. And I've learned that the key to fast execution is having depth in the different domains that come together in that product. And lastly, first principles driven approach is the fastest way to do product development, however counterintuitive it may seem. So anyway, with that, this is just the running theme across the presentation, and you'll see reflections of these specifics as I talk about the story. Okay. So I think I'll spend most time on this point of care diagnostic system. I spent a lot of the day talking to people who are interested in point of care diagnostics. I think probably that will be more interesting than other things. So I'll spend most time on that. And it has this razor blade model where there's a disposable and a durable. Then I'll touch upon the two wearable drug delivery products that I've worked on. One using needle-free injector and one using dissolvable microneedles. And then I'll close with the story on engineering surfaces. And finally, I'll just mention this work that we did at Iit Bombay, which actually was all optimized for learning, not for productivity. So anyway, so that's the outline. So let's dive into diagnostics. The goal was really simple. Like most of us who are working on point of care diagnostics, imagine that anybody should be able to do the test, and they should be able to do it when and where it's needed. So you're trying to get the lab quality results in field settings, right? So that's the running theme. So that's not unique in that sense. But yeah, so that's what we also aim to do. And I thought a good way to understand what we were doing is to just see how the product was intended to be used. So basically, it's a shoebox size analyzer with a touch screen. And this is to be used in a pain management clinic. And what's really happening is the operator is handing over the cartridge, which is also an oral fluid collection device. Maybe I should pause here. So as you can see, there are two prongs. They are absorbing oral fluid. And one side is collecting fluid for point of care testing, and the other side is collecting it for the optional lab send out. So if the doctor so wishes, he or she can send it out to the lab for a confirmation testing. That was the idea. And it has molded parts, it has a TPE membrane, there is a blister pack, there is a biosensor. So it's these porous media, this is sintered plastic parts. So it has all kinds of parts that have come together to create the cartridge. But the heart is the photonic biosensor chip. Now there's a change in color that indicates that sufficient saliva has been collected. And this sheer act of capping pushes a buffer through the porous media, causing the saliva to mix and transfers it to the cartridge. And this is just scanning QR codes to tie the cartridge to a patient, right? And at that point, all that the operator needs to do is insert the cartridge, touch the start button and wait. And while this wait is on, the reader controls the fluidics, and we will spend a few minutes looking at how this works. And basically, the fluid is being sent after mixing it with reagents to a photonic biosensor chip, where lasers are probing these binding sites, and the fluorescence is being collected and being sent back to the reader. And in 15 minutes, 10 classes of drugs are detected. So the idea is you detect it while the patient is still in the clinic so that the doctor can make a decision based on the test. So that was the use case. Right, so that's the Diagnostics product. And why was that needed? Well, you probably have heard about the opioid crisis. Given the COVID-19 pandemic, we have not been talking as much about the opioid crisis, but the truth is that this pandemic has made the epidemic worse. There's been 70% more people dying because of the pandemic. And after speaking with about 50 plus people across the US, what we learned was medication-assisted treatment was the most effective way of saving lives. And that required a point-of-care testing in the clinic. So they did need a screening solution, something which they could get answers to in a short time. So that's how we landed on the product that we came up with. And I'm a stickler for process. Maybe being a chemical engineer, this comes easier. But if you follow good processes, they don't guarantee you great outcomes. But the converse is very true. If you don't follow great processes, it's almost certain you will not have the right outcome. So I think I love being a maven for these following good processes. And Stanford Biodesign process is one such thing. So basically, what they emphasize on is you spend time identifying needs before you go to invent. You don't want to make a great thing that nobody needs. So I think having this discipline of iterating and finding the needs, prioritizing the needs, and even if you don't do everything well, whatever you choose to do it well enough so that it's meaningful. So that's what the emphasis is, and that's what Steve Jobs has been teaching us. So same thing. So then you move on to the invent phase, where you're going from you go wide, you do zoom out, and then you do zoom in. That's what you do at all levels. So you're generating wide ranges of concepts, then you cull them out and pick something. And before you go too far, you want to get feedback from your end users. So implement it in the field and let them tell you why it's not good. So that, again, you don't want to be spending a lot of time, money, energy developing things that may not be valued by a customer. So that's it. I mean, it's a very commonsensical thing, but I thought I should say this. So when you do that, for this use case, what we found out was urine collection was such a nuisance because for drugs of abuse patients, you're talking about for pain management clinics, they do same gender observed collection, which is fun for neither the collector nor for the patient. Then if you don't do that, the sample is prone to adulteration. And then they're trying to read and guess the color change, which is never a great thing because there's ambiguity. And finally, not all tests were available at point of care, so they had to send it out to the lab, which meant a wait for a few days, which adds to the cost. And ultimately, the cost gets passed on to the patient. So easy observed fluid collection that eliminates risk of adulteration was the most important thing. And clear objective readout results in 15 minutes. And basically, you collect 2 samples simultaneously so that if the doctor wants, that option is there with them. So with that clarity of needs, we developed this product. And these were the 10 classes of drugs that it looks at. Most of them are things that no patient should be taking, and few of them are things that are prescribed as medication. So that's that. And these also, we met with doctors, toxicologists to arrive at what should the panel be and what should the cutoffs be. And yeah, I'll keep using the word robust because, when we invent something, we get so excited when something works for the first time in the lab. And here, if I look at my iPhone and it doesn't unlock, it's very disappointing. So you want something to work every single time. You don't want to think about it. So graduating to that from a concept is the journey. And that's what I mean by robust. So all the design choices are guided by that thought. There are many cuter things that one could do, but you don't want to do that. You would rather have something robust. That was the priority. So yeah, so basically what I needed to lead was there is a system of product development, which is what are you making? And I keep likening the chemistry is like the app on an iPhone. You develop the hardware and then you port the chemistry on it. So that's what a particular assay is. Yeah. And once you know what to make, then the how to make, which is the process. And then you go to automating parts of it. And we didn't go to all the way commercial, but along the way you develop new IP. I don't know. Okay. Sorry. So, I want to spend a few minutes on this slide because everything that we were able to pull off was because we had a great team. And why do I say this is a great team? Because we complemented each other's strengths really well. So when I started, I knew that I needed someone who understood photonics really, really well. So the key person was finding David Pileup. Now, David is an undergrad and PhD in physics from Berkeley, and he can see photons move. I mean, having 200 patents is one thing, but he actually understands physics like very few people do. And so getting him on board, getting him excited about how cool this project is, why it's worth doing, and why he'll have a lot of fun working with us was important. And once we had David on board and he agreed to do all the photon budgeting and modeling, then I needed an experimentalist and somebody who could create a good team with David. So that's why I hired Hardeep out of Stanford. He's a PhD in optical physics from Stanford, but his physics is all right. What he's really good at is analog electronics design. And we needed that. And plus, of course, he could do experiments as well as anybody. So two of them formed a great team, right? And then for the disposable design, I bumped into Pranav. Dr. Premnath knows Pranav too. So Pranav is an undergrad and master's in mechanical engineering from IIT Chennai. He did his second master's from Cambridge, worked for Sagentia for about 6 years, and he was at Stanford Biodesign. And I hired him only for 4 months, but 5 years later, he continued to be the lead architect. Fantastic person and a very good mechanical engineer, designer, and he understood microfluidics reasonably well. And that was very important, somebody who combined both mechanical design and microfluidics. Now, like I say, design you can outsource, but characterization and troubleshooting, you still have to do yourself. So for that, I had Suklin, and she is a BITS Pilani chemical engineer from BITS Pilani, and she did a master's from UC Davis. And there were many bigger brands, right? Like there was somebody from Berkeley and Intel, somebody from Stanford, but really who was good for that? I felt that Suklin was just fantastic. And she created a great team with Pranav because Pranav could design and she could characterize. Similarly, Yunpei, I mean, I had a chemistry PhD from Caltech, but those are very one-dimensional. But I needed somebody who understood chemistry, but also understood enough about optics to be able to troubleshoot when there are problems. And Yunpei had her PhD in biophysics from Berkeley, and her undergrad is in physics. So that helped a lot. She had picked up all the biology and chemistry in her PhD. So she became the person who would put the assay onto the cartridge. And she again teamed really well with Pranav and Sukleen. And finally, I mean, I must emphasize Benton, who's a high school graduate, and I wouldn't trade him for any PhD because I felt that, you're only going to be effective in problem solving if you have good observations. And he could observe like few could. And he was almost meditative when he looked at a part. Anyway, I used to say that he had very calibrated vision. He could say you're off by so many thou. So anyway, so the Benton. And for all of us who are on a regulatory path, I would urge you to find a very good regulatory person who understands science also. So Anna is a JD and she was an SVP in a few companies, but she understood science really well. I mean, it helped that she had a master's in physical chemistry from Stanford. And so you need people who are really good at what they do because you don't have redundancy in a small team. I don't have a team of 70 people. So every person, I mean, I used to say we are only as good as whoever doesn't do his or her job. So there is no poor, I mean, anyway, so that was that. And just another note on when you're selecting vendors, I had many big companies in the US, but I selected JB. And he has a company in Bangalore. He's a Iit Bombay undergrad and did his master's from Stanford. His 70 people company doing automation in Bangalore. He did actually automation for the Ninjas company too. Yeah. And so bottom line is it's much better to have somebody who may not have a big portfolio, but is hungry and committed to excel in your project. I mean, that is much more value to me than the big resume. So, and that turned out to be a great decision because I think JB was as good as anybody. So I felt, well, with this team, we were as good as anybody in the world. We were second to absolutely nobody. And I feel having that team was the difference between when we could have not done anything, but the reason we were able to do was because of the team. And most importantly, we all stuck together for 5 years. So anyway, so what was I doing if everybody was doing everything? So yeah, what happens is every time there are these deep domain experts, interfaces can be orphans. So somebody needs to be watching the interfaces like a hawk, and that becomes my job. So anyway, and that's a lot of fun because you need to understand a few things well to do good troubleshooting and make good design choices. So again, let's zoom out. There are 3 things flowing in the system. One is the material, the saliva flows, it mixes with antibodies that are tagged with fluorophores that are being dispensed on the biosensor chip where a binding reaction happens. And the kinetic assay is being performed, light is being measured and reported. So the moment light is being measured, the second thing that's flowing is light. Laser comes in, excites the fluorophore, and the fluorescent light travels back to the reader. And once you convert the light into an electrical signal, that's where, and then that signal becomes data, right? So then the third thing that's flowing is data. And these days, everything is an IoT. Something has to go to the cloud. So yeah, that data gets uploaded in the cloud and all that. So there are 3 things flowing, right? So what I'll do is give you a flavor of a few problems in each of these modules, something that happened here, something that happened there, kind of thing. So yeah, as you know by now, the test cartridge has collection, assay, fluidics, and biosensor. And analyzer is really the controller for all of that. So, here is the chip has waveguides, and the good news is that this is all very trailing edge of technology. We are talking about tens of microns dimensions, right? You're not talking nanometers. So there are many fabs that are able to do this. And you're able to, so anyway, so that's how the waveguides were made. So it's just thermally grown oxide. Then you have LPCVD nitride. Then you clad it with PCVD oxide. Then open up windows in oxide and then put ALD oxide, which is nanometers, on the surface. Why are we putting the last oxide layer? Because ultimately we want to put biomolecules, and the chemistry that you need to do requires oxide on top. So that chemistry is the fab doesn't do for you, that you have to develop yourself. So that we develop that chemistry using how to do the silanization so that you have a hydrophobic coat on top. And then on top of that, we deposit these 32 spots. Each of them can potentially be a different biomolecule. But in this case, we had some redundancy. So some positive controls, some negative controls, and some redundancy because we have only 10 analytes that we are looking at. So, but when I'm just showing you one slide, but this is a lot of work, right? And then what happens is you don't want to do singulation first. And then if you do printing, then that you've lost the whole power of wafer processing. The whole fun is that you can do several thousand chips on a wafer at a time. If you do singulation first, which is the conversation we were having earlier in the day, then you really don't benefit. You're now introduced sources of variability that were avoidable. And what happens is in a lot of product development, you start off talking about means, then you only talk of variances. Your enemy is your variability, and you're trying to clamp down on that variability from everywhere. So anytime you see an opportunity to eliminate variability, take it. Right? So that's that. So how did we, once you have deposited biomolecules, you can't use a regular salt dyeing and dunk it with a coolant. So you needed to have a different technique. So Hamamatsu makes a laser with which you can do laser dicing. So it creates stresses and doesn't heat up the biomolecule. So you're able to do the singulation or dicing without bothering the biomolecules. So basically we went from handling single chips to coupons to full wafers. And that is what I was talking about, scalable manufacturing. What you see on the left is not scalable. What you see on the right is scalable. Right? And that is true for every element of it. That's all. We're getting deep into okay, so what is happening here at the sensor? Well, think of it like light is emanating from the surface, and it's that evanescent light that's exciting the fluorophore. So if you think of this table as the well, the light only extends maybe this distance. It's very close to the surface. The rest of the chamber is all dark. So all fluorophores that are floating are not even excited. So you're not collecting any fluorescence back, which is very smart because all non-binding fluorophores are noise and everything that is bound is signal. So if you look at the concentrations we were able to detect, we are able to get to 13 nanomolar concentration with non-binding, but 2.26 picomolar with binding. What it means is the signal by noise ratio is roughly 5,000. So I think, just making a point that, see, good engineering can make you leverage, get the whole impact of good physics. But if there is no good physics to start with, any amount of engineering is not going to make it any better. So I think when you're doing due diligence, or anyway, when you're establishing the boundaries of any technology, so I needed to understand this really well and make sure that our use case, this was good enough. Right? So that was that. And by the way, those of you who probably, many of you know what evanescent wave is, but all of us have done particle in a box. And then there is that wave function that extends outside the box. So it's like an exponentially decaying wave, that's why it's confined to very near the surface and everything else remains dark. And this is what I was referring to as downloading the app. So we had created such a robust process for porting assays because there were 10 different assays. So you had to have a method in place that, okay, how do I develop an assay? How do I screen reagents? And how do I put them on the chip. So we had a method for that, because every step is adding more variability. So you need to select the right reagents. I mean, I think I'm repeating, so belabor that. And yeah, so selecting the right assay format was the key. Then we went on to do a two-step assay. So I'll just briefly, what's happening when the control solution is binding, there is a lot of antibody in it. And therefore, you get a slope that's proportional to concentration. But once you let the sample solution on the chip, most of the antibody is lost because it has already been used up by the analyte. So the remainder antibody is lower in concentration, therefore you measure a lower slope. And it's the ratio of the control slope and the sample slope that is your assay signal in this case. Right. So I think, again, there are a few innovations. These are all three of them are there to fight variability. All three of them are improving if I do 20 cartridges, each of them is slightly different. And I want to do some signal processing such that I'm reducing the impact this variability has on me. So that's what all this is. And I will skip the maths because it's relatively straightforward, but not all that relevant. But I'll spend a few moments on this slide. You know, this rigor and creativity is a false dichotomy. You need to be both creative but rigorous. And what I mean by that is when you're talking about, oh, this is my design input. So you need to be very clear, this is the design input, and or this is the problem, this is the root cause. This is the hypothesis for the root cause. I haven't yet verified it. Okay, I'll do this testing and now I'm sure this is my root cause. And now, oh, there are these 5 design choices that can address it. And I'm selecting this and then I'm verifying that. It's a very methodical thing. You don't want somebody who's describing an observation who's suddenly telling you about what solutions may be good. So when you're at this column, don't go to the next column is the kind of discipline we need to have. Otherwise, in a big group, I think it's more chaos and it's tough to get things like move is what I learned. So that's one of the values, having a disciplined approach with a very light QMS can help you do. So when we say cartridge works, it does so many different things, right? It collected metered quantities of 2 saliva specimens. It does mixing, it does transfer. Then there's a metering, it's measuring 20 microliters. Then it's mixing, then it's incubating, and finally the assay. So each step needs to have its own performance metrics, very cleanly articulated. And then each step needs to have how all can this fail? What are the signatures of those failure modes need to be very clearly understood. So I think that's where the time goes, right? You need to have that clarity right through across the system. So that's what we did here too. And I'm a huge fan of being modular development, meaning when one unit is doing 6 things, it's really 6 units combined. So you need to be prototyping those 6 things separately, making sure they're working as they should, and then stitch them together. Otherwise, this doing troubleshooting is near impossible. So again, I'm not saying anything that's not obvious, but I think I'm just surprised how many times people don't do it. So this is the innovation bit. So yeah, this is like 3 valves. We wanted all fluid to stay confined in the cartridge. And basically, just by turning on and off, okay, so basically, by opening and closing these valves, one is doing metering, mixing, incubating of the 2 fluid slugs. Right? Use a mouse. Yeah, people will miss. Yeah, sorry, sir. Yeah, so basically, just by opening and closing these valves, one is able to handle 2 fluid slugs. And first you put 1 fluid slug on the chip, then after that you put the second fluid slug. So that's what the entire run is. And all the time, it was designed to keep the fluid confined in the cartridge because that was a safety thing. And yeah, I mean, we started with machine prototypes, then went to molding. And there's no real substitute for this iterative things. You can be smart and have fewer iterations, but be prepared to do a few iterations is what I say. We only had to do the second molding iteration to get to the minimum viable product, which I think is like pretty decent. But you need to be prepared for that is all that I'm saying here. And again, you have to keep an eye on the risk, and what risk is acceptable and what is not is a judgment call that you're making at every design decision. And along the way, you are solving problems. And on micro scale, mixing is non-trivial. So where needed, we did COMSOL multiphysics simulations, and then we used the chip to probe the degree of mixing. So if you look at this plot on the left, there is no analyte here and a lot of analyte here, which is not good because you want the slope to be uniform, which is telling you that the concentration of the analyte is distributed uniformly along the length of the slug. So when the mixing worked right, we had what is shown on the right. So basically using your own system as your debugging tool. Again, non-specific binding, leaks and bubbles are the bane of many, many microfluidics products. So nothing unique here. We had to fight non-specific binding. So we came up with a plasma activation and coating process that solved the problem. I mean, nothing really special. Yeah. Yeah, thanks. So yeah, it had all kinds of parts, as you can see. And all these parts are made by different vendors. There was molders to make molded parts. There's silicon chip at the fab, which had the post-processing we needed to do. This is PSA, which is die cut. We started with laser cutting, graduated to die cut. The porous media is a sintered plastic part. So there are different vendors, right? But then the assembly was done by us. And by the time, we wrapped up this operation, we had built and tested about 13,000 cartridges. And basically, yeah, and you had to have low enough failure. Otherwise, again, it's not the right design. So, and once we had this manual cartridge assembly, only then we graduated to automation. And here also, you rank the risks and the red, orange, and green. And green I can carry on, but the reds and the oranges needed to be solved. So those were the first ones to be automated. And even there, basically we did a prototype for automating in Bangalore before we built the actual equipment and shipped it. So, right, this is just some mock-up. And here I was telling you everything about the cartridge, but the reader also evolved, right? We started with no fluidics. It's only chip and optics because that was the foundation of everything. Biosensor was the heart of it. And then I said, basically, then you add the plumbing, which is microfluidics. And then everything in this blue box was functional, but not really ready for scale, because this is all using very expensive components and lab view things. We transitioned to everything Windows and something that could be built at scale, which was this. And we stopped here for this product, but we prototyped the handheld as well, because there were some other use cases that would require a handheld. For the software too, there were things that we bought off the shelf. There were things that were dependent on the hardware. And then there were things which were totally independent of the hardware. I think to me, analytics layer was the really crucial one, because we could identify different kinds of failure modes. And that was so important. When you're testing thousands of cartridges, you need to know the frequency of occurrence of each kind of failure mode. And so if all that is built into your software, then life is that much easier. This was really satisfying to go to the clinic and conduct this study. And 335 out of 342 results were right. This was more than what I thought would be for the first study. And even more satisfying was the response that I received from the doctors and patients. Some of the doctors said that, why don't you keep this here? I said, no, we need to take it back. So it was all good. These are the investors. And, it's good to have people who may not understand what's going on at all, but they'll ask you questions that are still meaningful. And I think not valuing those questions too is a good way to keep yourself honest, is what I learned in the process. And some of them are very, Mickey was a senior vice president in Siemens, as was Jerry. They've run their own companies. They were leading 10,000 people teams in these big companies. So they know a lot about chemistry and engineering. So it's good to have somebody who's asking me questions every few months. So I think that was helpful in many ways. And again, regulations are not optional. They just need to be followed. So they have to be baked into your manual, your key SOPs, and so on. So that was that. I'll just talk about two products here. This, we went from concept to stage 1 and phase 2 clinical studies, and then the company went public, and that's when I quit Corium. But it was really cool to develop these microneedles that are made up of sugar. And we partnered with Pfizer, GSK, Novartis. So everybody had their own API. So in that sense, this is a platform, and you can have different kinds of drugs being delivered. And the needles go in, and it's the moisture in the skin that dissolves them. So what comes out is really no sharps remaining. So it's a room temperature stable microneedles patch, which can be self-administered painlessly. So I thought this was really cool. But the make or break was scalable manufacturing. And again, it was a highly multidisciplinary project, which required chemi and ME in equal measure. We won the Edison Award for this wearable patch pump. Here we are using the momentum of the liquid to pierce the skin and act as a needle. So it's just like an inkjet printer using a piezo crystal to create the pressure pulses. And the nanoliter pulses are being deposited in the epidermis. That's what this product was. So I think this was all cool, but this really didn't go all the way because of the recession. I'll switch gears and give you a flair for just a flavor of the inventions, which have also been a lot of fun, and some insight into why surface physics is so cool. And all of us who are working with biosensors and microfluidics know that surface is dominant. In academia, these become the metrics, right? The papers. So yeah, these papers have been cited about 300 times each. So they were well received. So really all microfluidics is really, there's a chamber, liquid comes in, liquid goes out, and there are sensors. They are either electrical or optical. So we said, why don't we flip the paradigm and say, okay, there's a bed of sensors and I can move a droplet on them, right? And you can do metering, mixing, because drop is like a quant of liquid, and then you can merge them, mix them, do whatever. And now this has become the norm in some areas of diagnostics. And after Raindance Technologies, which has been acquired by Biorad, droplet microfluidics is the way to do fast assay. So, but this was early in that story. And how do droplets move on surfaces? Well, just like a ball goes down an inclined plane, it's an energy gradient that moves the droplet. Right? It's a surface energy gradient. So people were looking at different surface energies. There was a Richard Fair's group in Duke. They were looking at electrowetting to create these gradients. Later, a company called Advanced Liquid Logic came out of this group and was acquired by Lumina for 96 million. This is Whiteside's group. He's creating surface assembly, basically chemical gradients, and showing a gimmick that a drop is moving uphill. This is Sandra Trojan's group. What they are doing she was at Princeton, now she's at Caltech. What they're really showing is if you create temperature gradients, surface tension is a function of temperature. So you have also created surface tension gradients, and that can cause the fluid to move, something called Marangoni effect. So that's what they're exploiting. So I looked at all this and I said, okay, why don't we engineer texture so that there is an energy gradient because of texture and the drop should move. So that's what I did here. And okay, I should show these two videos. Basically, if there is a gradient and if you vibrate a surface and you place a drop on it, the drop starts moving. And they're very simple, nothing. And if there are two gradients oriented in two opposite directions, and you vibrate, the two drops go into opposite directions. So that's also easy. So now I said, okay, but the problem with these gradients is if you want a very long track, it'll be a very small gradient. And you can never close a circle with a gradient, right? So I didn't like that. So I thought, okay, what can we do? So then I started looking at ratchets. And ratchets have been around for a while. Robert Brown of Brownian motion fame, he introduced ratchets right then. And he, in that year, I don't know, whatever he was doing, he did everything that was to be done. So including modeling ratchets. And Perrin investigated them with experiments, the ratchets. He may not be very famous. Some of you may not know him, but he's also a Nobel laureate in physics. And the reason I was exposed to all this was only because of Feynman, because he made it, his lectures made it to every home almost. So what Feynman said was really, if you have Brownian motion, you can make a flywheel move. And by the way, you are not violating any thermodynamics. Why? Because you are expending energy in moving this ratchet and preventing the flywheel from moving in one direction. It's exactly like the flywheel of the bicycle. There is something that's preventing movement in one direction. So it's only allowed in one direction. So when molecules hit it, it moves in one direction, but it's not allowed to move back. So I thought, can I take this idea and transfer it to the surfaces? Right. So that was the inspiration. And also, when you look at surfaces, this is a lotus leaf surface. And what happens is, see, this is almost like a ball. It's like a point contact. And why? Because if you look at it with an SEM, that's what the lotus leaf looks like. It's a bunch of pillars. So the wax is the same wax, which will not be quite as hydrophobic. But because it's these pillars, the drop is really resting on pillar tops. This is just some math because I needed to design these surfaces. So it's really like a fakir lying on a bed of nails. The drop is resting on pillar tops. And when you push the drop, the energy of the system goes up and then it plummets to what is called the Wenzel state. So I thought, let me recreate this in silicon. So I created surfaces, put drops and saw the drop resting on pillar tops, pushed it and it went down. All this is straightforward, good. But then the insight was, if I vibrate it, the advancing of the drops doesn't care about texture, but the receding of the drop cares about texture. That was not obvious, but that was something I observed. And basically that became the reason why I designed surfaces that look like this. So if you look at the right edge, it is contacting many, many pillars. The left edge is not contacting many pillars. So when the drop expands, it expands equally. But when it's trying to shrink, the right edge stays pinned, the left edge moves forward. So the entire drop translates. So what we're really saying is you can create local asymmetry to cause global movement. So that, I mean, then this was all hypothesis. I mounted these surfaces on a speaker and vibrated it and all that. And yeah, I mean, everything that I thought should happen did happen. And the drop started moving in circles. And because Whitesides had done this gimmick, I thought I'll at least repeat it. So make a drop climb uphill, etc. Right. So anyway, so that was that. So and you know what, in non-specific binding, we always lose protein in any bioassay. So I thought, okay, you have peg-like surfaces that prevent fouling. Can you have a hydrophobic surface which is also non-fouling? So again, short of it is that I created surfaces where the tops were SAM or the tops were PEG and the valleys were all hydrophobic. And when I placed a droplet, in spite of the droplet contacting only silic surface, it's having a very, very high contact angle, as if, because it's really because of the pillar structure. That's all that is cool. So anyway, I'll switch, and my last couple of slides, again, I'll start by acknowledging my team. This is my mentees at Iit Bombay, and because of them, we could do what we did at IIT. And when I went to Stanford NanoFab, it became so easy. You push buttons and things work. IIT, nothing worked. So I felt that it was optimized for learning, not for productivity. The product were we, not the silicon wafer. So anyway, so because nothing worked, we learned a lot, and so did all these kids. And Anirudh went on to do his PhD from MIT in EE, did his postdoc at Harvard, and now he's a faculty at Georgia Tech. Now, Siddharth, he did his BTech, and then he took up a job with McKinsey. But when I was in UW, University of Washington, he came back to do his master's. Halfway through his master's in EE, he shaved his head and all and decided, now I'm going to be a physics guy. Anyway, so good news is he did get into MIT and Berkeley. He rejected both. He ended up accepting Princeton theoretical physics. He did his PhD in biophysics. He's a faculty in Toronto right now. Bhaskar was the most laid back. So he ended up doing his PhD from Ann Arbor. He's a faculty in IIT Delhi right now. So all three did all right. And unless we all worked together, we couldn't have pulled this through. So, what is this? Yeah, we were really trying to look at complete electrical system where you are pulsing to detect capacitance dips because of DNA coming, different DNA slugs as they moved past the electrode, they created a dip in capacitance. So that was one project. And the other one was creating this aperture in silicon so that we could then flow a suspension of cells through this. And what's happening is as cells flow through, a cell is really a dielectric shell, and it's the conducting medium that's letting the current pass. So as this dielectric shell obstructs, you are increasing the impedance, and if you are probing it with a constant current, you are measuring a higher voltage as you obstruct more and more. So if it's a tapered thing, so that's what is happening. So we detect a cell, size it based on how much change is occurring, and based on the size, you apply a pulse that will not kill it, that will not lyse it, but make it permeable enough so that DNA can go in. So that's what we did. We counted, we sized, and we purated it. And the bottom line is that we were able to get, this was early results when we got 89% viability and loading efficiency of 98%. But both of them, we were able to up to 98%. And that's a paper that's also out. So it wasn't cool then. Now it's very cool because this can be a tool for inserting genes into a cell for gene and cell therapy. So, okay, where did I do all my education? I mean, I thought I'll just end with 2 slides on my background. Yeah, I learned to think at IIT Bombay. And yeah, it's chemical engineering and microelectronics. And later, when there was a choice between Cornell, Berkeley, and UW, University of Washington, my reason to go there was because they had money and didn't know what to do with it. So that's a good situation where, so again, you need to have that clarity. Why are you going there? So anyway, and this is the extracurricular activities I do like to help in if you know things and you are useful to nobody, then by definition you're useless. So you don't want to be that. So that's my way of helping, that I do serve as the reviewer for SBR panels for NIH and NSF. I also served as the industrial advisory board member at UCLA. And EPIC, we organize these conferences to bring engineering and life sciences folks together, because that's the interface which is happening today. And I'm also a senior member of IEEE and serve on technical program committees a few times. So, thank you.
Thank you very much. So, Ashutosh, I think I'll start the questions and then I think Pragya has some. Yeah, that's okay. This is also here, right? So, a couple of things. The first thing I wanted to ask you was, all these multidisciplinary teams have all these different people with specific expertise, and you said that yeah, your role is a an interface. But before that, somebody has to conceptualize a big picture, right? Where all those people fit in, right? So can you talk through that a little bit? How did you go about constructing the, for example,
Diagnostic team.
Diagnostics, you must be who conceptualize the big picture of what needs to be done. How is that idea conceived? How is the problem defined? How did you come up? How was the solution conceived? Then you can put the pieces together, right? So that process, if you can walk us through a little bit, broadly, without the details of the science. I just mean in terms of, how do you go about picking the problem, defining it, and
So, when I present, it seems very logical, but actually it's very chaotic the way it actually unfolded. So, because there are different people with different opinions, right? So, Index Life Care was not even looking at oral fluid. They were looking at blood and cardiac biomarkers. That's a very crowded space. And I didn't know that we were very differentiated in that. So that was one, but that's where we were. And we happened to have an investor who are one of the top toxicology labs in the US. And their interest was oral fluid because they had been watching the field for a while and they felt that oral fluid is poised for taking off. And there are many, many advantages. See, that perspective I did not have. I got it only because of the toxicologists who had been watching the field for a while and they were convinced that that's the direction to go. We looked at two products then. One was we looked at a few products, few use cases. One of them was all about police car and roadside testing.
yeah,
right? Because that becomes very easily available. But the problem is that technically that is more challenging because the robustness required, the ruggedness requirement is much higher there. An AC room like this.
So the first problem must have been finding substances in saliva for, say, people who are driving under the influence of some drugs or something like that. That must have been the first motivation, right?
Correct.
So, okay, so it was clear that you had to use, say, saliva as a body fluid and look for certain markers, right?
Correct.
And then you start.
Not markers, these small molecules directly.
Small molecules.
You're looking at THC, you're looking at heroin, whatever.
Yeah. So now there could be multiple methods to do that, right? Solutions to do that. Why would you pick this, and how did you think to that?
See, some of it's a hammer looking for a nail kind of thing too. I mean, that's not how it should be, but that's the technology we had in hand.
Yeah.
And then the question wasn't what is the best technology? Can we make this work for this application? Was the question we were answering. And for that, the limit of detection thing that I was telling you, I wanted to convince myself that the cutoffs that are needed are much higher than what the platform is capable of. So we did early experiments with streptavidin, biotin like thing, where I know that the K-off is near zero. So you want to establish that if there is nothing coming off, how low can you go? And that turned to be much lower than anything we needed. So as long as we had the right reagents and the K-on and K-off were right, then we would be able to do the job. So that's how I convinced myself. Rest was engineering.
Yeah, but you were convinced that you could get the necessary sensitivity and
Correct.
specificity that you wanted.
See, sensitivity.
Time, cost.
Yeah, but see, sensitivity is a function of platform. Specificity is a function of reagents.
Yeah.
Reagents just need to provide the specificity. That platform cannot provide. So yeah, so that we understood that basically how much effort is needed for doing a 10-plex assay. So that people were freaked out about that there'll be lots of interferences. What happens when you do 10 of them together? So there was some risk that we had no answers to. We just had to do it.
Yeah. So what about time of doing this detection? What about the cost of doing it? Does cost figure very early in the tech development activity? Do you consider cost as a factor when
Yes.
you are looking at that, or would it come much later? Because first you focus on sensitivity and specificity.
So, it's a parallel thing. While we were, I said, you can't, you're hiring people, you have no time to lose. So you have to start doing something while some things are being figured out. So in some sense, you are doing this parallelly. But in the needs finding exercise or the product requirements document, the cost is early.
Okay.
You need to have very clear cost targets.
So in such products, regulation is quite important, right? So at what point did regulation kick in? How did you plan for regulation? I mean, did it affect your design? Did it affect your early experiments? Did it affect your studies you had planned? At what point does it kick in? And I also saw some elements of QMS in your slides of risk management and so on and so forth. So when does that kick in? Because it seems very unreasonable to think that it starts from day one. I mean, practice, I suppose.
Right. See, so there are two parts to it. One is creating the documents. One is learning to think in that disciplined way. So if you get busy creating documents before the team has actually imbibed how to think about it, so then we are wasting time. So I wanted to I had this QMS light where you don't want to enforce any of this documentation, but a lot of
The principles it brings to the table, you want to adopt.
correct.
Right?
Yeah. And there is some learning curve because many of these people are doing all this for the first time. So a few of us who had exposure, I was one, Pranav was one, we became the people who were like disseminating, etc. But so we had a QMS Lite where I just had matrix, simple Excel sheets.
How much do you need to be an expert in the regulatory process or the scale up and manufacturing process? Because I suppose your primary interest will be in making sure that the science works. But how much of an expert do you have to become in, say, manufacturing, scale up, regulations?
See, scale up and manufacturing, I treat them everything up to pilot manufacturing lies squarely with R&D. So it's if I'm doing that role of the CTO, then that's very much on my plate. I need to take it to a point where our job is to make ourselves unnecessary, right? So a team of this caliber should not be needed. After that, a set of instructions and anybody anywhere on the globe should be able to make that. So taking it to that point is totally this team's responsibility. But as regards regulatory, there is nothing special in terms of regulatory. It's only the study designs that are guided by the regulatory path. But quality.
So like, for example, you mentioned that certain things which were red, you have to plan for automation, right? And that seems to be also a requirement of the regulators, right? So,
but I'm not thinking of regulators at that point, right? That's the normal product and process development methodology that we've adopted. We always take on the highest risk things first, right? Irrespective of what the regulators want. But the good news is, if you do it this way, then it's easy to have that conversation with the regulators. So when we had our first meetings with the FDA, it was very positive. See, they can't tell you you're doing great, but if they're not saying anything negative, that's a good thing.
And are they open to discussions?
Oh, yeah.
US FDA
Yes.
open to giving you comments
Yes.
and pointing in the right direction?
Not just open, they want to do that. And they see, if you postpone the first conversations with FDA till the very end, it's good for nobody. So they also want early engagement, and they would nudge you in the directions that they feel is reasonable. And I see, with 3 products that I have had to deal with FDA, I feel that if you do solid science, more often than not, you will not find anything special that you need to do for FDA.
Okay. My just one last question before Pragnya, I'll ask her to ask those questions which are on the chat box. So how important, can you comment on how you went about thinking IP and IP portfolios for your technology?
Yeah.
And again, how much do you need to know and how much help do you need to take from others, right?
See, IP is a purely defensive thing, meaning I don't want to be stopped because I'm infringing on somebody. That was my bigger concern. And so anything that I feel is novel, so we kept filing the patents for. And I actually went to the USPTO and had an one-on-one session with the examiner, and that made our subsequent 5 patents so easy. Because the lawyer did all the talking, but he did not understand much. And from his questions, it became apparent to me that he hasn't understood. So I told the lawyer that let's both go to DC. And we went to the USPTO office, got an appointment, sat in front of him. And he said, can I hold on to this cartridge? I said, great, please do. So I knew that the next patents would also come through without questions, and they did. So as regards, when you start working with outsiders and you start sharing the process details, that's another time where you need to think about patents. Because when you're working with, say, Harro Hofliger of Germany, we worked with for some design, their whole game is IP. They want to protect everything. So I need to have very clear things of what is yours, what is ours is ours, and what we do together we'll figure out. So then before I'm formally engaging with them, I make sure that all my process know-how is protected. Normally, I don't like to disclose a lot of process. All the patents are usually product patents, meaning you want to tell them what it is. You don't want to teach the world how to do it. You want to tell what and the results.
Yeah. Okay. Pragya, you want to take a couple of questions?
Yeah. We have a question from Avinash. Do you have clinical data that support that detection concentration of drugs in blood and saliva are comparable?
Right. So, blood, saliva, and urine, all 3 have different concentrations. And if you look at the time window, so yeah, that is not the comparison that is needed or relevant, is what I'm saying, really, because they give you slightly different information. So oral fluid and blood are still somewhat correlated, but again, I mean, we can do it for curiosity, but it's not something that is needed for anything.
So for a given diagnostic application, you might just be interested in, say, what some number you're getting from the saliva and whether it matches with some reference method, not necessarily that it matches with blood.
Correct.
Right? That's
because that work has already been done.
Yeah.
See, if it was a very new thing that we are doing something for the first time, maybe that was necessary, but that is not the novelty that we are doing today for the first time. So that's why we didn't need to do that.
Okay, another question, Pragya?
Yeah, another question we have is from Dr. Girish Kulkarni. How did you address large-scale manufacturing of such intricate and complex diagnostics? Example at Evanostics.
See, because some of the things were so unique to what we were doing, we chose to build the pilot equipment and do the pilot manufacturing ourselves. If it was I didn't want to give it to Hochuen in China to do it for that reason. Because what happens is, and even before in Korea, we designed the pilot line. I called it a pre-pilot line. We had all those things working right before we went to Harro Hofliger to do the next level of automation. Why? Because like I said, for these complex things, there's so much learning that happens when you do it that you don't want, basically you want your product development team right next to where all this manufacturing happens, because they are in the best position to do the troubleshooting.
Yeah. Okay, any other questions?
No, that's it.
Any questions from the audience here? Anybody wants to ask any questions? Somebody was trying to ask a question. Somebody was trying to unmute, right?
Yeah.
Yeah, it's okay. Anybody has a question can raise their hand, please. Or anybody here has a question? Maybe the audience here. Okay, so I think if there are no other questions, we'll take it to a close. I just wanted,
I thought there would be some there.
yeah.
So these cartridges, you test them on in your factory and they are good, right?
There is some level
Ship
of
them. So how do we know when they are actually getting used, they are still in good state and still reliable and robust?
right.
Used?
Yeah, so we did all these tests, right? You ship it to across the coast and back and test them again before and after. That's how we knew that they can survive the shipping and handling.
But that's, what about the one which actually came to me and is bad?
See, so what you're really saying is there might be some intermittent failures and some may not work. So there is no real answer. You just have large enough numbers so that you detect those failures.
I'm comparing it to our digital chip design, right? Where we have to test our chips right when the car starts every time.
Correct.
And make sure that there are no failures.
So there is something called a system utility, what they call it. Basically, yeah, there is suitability tests that it does. There are some internal checks it does, but some you can detect, some you cannot, is how it is.
Correct.
At the start of the system, when the system is powered up, it is looking at a few things. And as the cartridge is inserted, it does look at a few things too before it starts anything.
Some basic sanity check.
And therefore, see, it's okay to abort a test, then give a wrong result. Right? So we have that built in. That there is a series of checks that if any of them fails, the test is aborted.
So you have like some level of quality control to the end.
Yeah.
So I have another question.
Yeah, please go ahead.
So it's actually an extension to the last question where you asked about how do you sort of like take up your idea and you make it as a product. When you make a singular product and when you mass producing the product, there are a lot of things that you need to sort of take, like you talk about the cost ratio and all of those things. So you talked about the engineering while you're building the product, but how do you sort of convert the mass production engine? Because that is also require a lot of intricate depths to it, right? So how, like, is it your specific team? If it's your specific team, like in your case, how did you work around that solution? Was it something different? How do you approach it? Because in the first scenario, you just needed it to work and give you accurate results. Now in the second scenario, you needed to work for a whole lot of systems. You needed to be economical. So that should also be like a, like an entire slide of 50 slides in itself, right?
Yeah. So basically, right now, this is only this level of work, right? Once you, like they say, if you're saying that what happens when you build thousands and all that, so maybe you will uncover new things, and I won't know right now.
I mean, you guys worked on automation, right?
Yes, we did.
So then, when you guys worked on automation, was there some parts that you had to cut out from the original product just to fit in?
Oh yeah. So design for manufacture. See, when you make these design for manufacturability changes, so I keep saying there is the design for performance, manufacturability, and usability. So manufacturability, automation is a big part of manufacturability. So yeah, there are some design changes made specifically to make the automation easier.
So it's like when you're nearing the end of the product, that's when you sort of like take a considerate effort that okay, now it should be also manufacturing.
Correct. No, not that late, yeah. So that's what I said, that you don't add manufacturability all at the end. But yeah, some of it happens later in the process. Some of it is thought through early.
And this is one last thing. It was related to the title of the seminar today. So you talked like I'll push it through the perspective of patents itself because we've already talked about it. So when somebody mentioned that you looked through the patents later on in the stages and like you went through, you made sure that you understood. That's like at a certain like when you've already achieved some milestone in your project. So when you talked about the ideation phase, so while you're actually ideating the idea, you're figuring out how to make it work. So obviously things align for you get a pass. But at that point of time, how do you sort of see you're not like infringing into, because it's not, I mean, personally, it won't be possible for you to actually see the multitude of research that's actually happening in the domain.
No, so we have these law firms, right? I have Wilson Sonsini is doing the work. So I asked them to do an FTO search.
But at that point of time, you don't have like a concrete idea. So how are you sure that like throughout the phase, you're not actually sort of like infringing over somebody else's idea?
So this is basically, you don't do it once and stop, right? At every something.
Sorry. Okay, continue.
Yeah, so at different points you do this, making sure that you're not infringing is an ongoing process.
So, and in case you come across something that you have infringed, or if it's on the borderlines, do you prefer to sort of like change the direction of your idea, or would you prefer to sort of like, like echo it, sort of
So-
rewrite for it? Like, what's more easier, what's more economical when you're designing a product?
So in this, in all three products that I talked about, we were able to make the changes so that we did not infringe on anybody.
So collaborative was more of an effort rather than just like diverting a technology to another.
Actually saying that you can work around it.
I could work around. So it was diverting.
That's what I'm asking. Is it more easier to work around in general cases?
No, that depends on a case.
It's a so case by case thing, right? So in these three, I can say that we were able to not infringe. We could make the changes so that we didn't infringe.
Thank you.
There will be cases where you will not be able to work around.
Right.
Okay, so then you have to, of course.
Then you talk to them.
But I mean, like, as a general principle, when you come across a case, but still, like, what's more easier? Like, what should you approach? Because
Yeah, so as a startup, at a very early stage, you may not even get the attention of the other party which owns the.
Correct. They don't come after you till you are till your stakes are high.
And they may not only come after you, they may not even be interested in talking to you.
Correct.
Even if you go with a request for collaboration. So most startups will first try to find a way to work within their needs, right? If they absolutely cannot, and then.
Yeah, it's such an see, Samsung and Apple can have that conversation. We as startups, it's not a level playing field.
Yeah. Okay, I think if you don't mind, we stop here. Thank you very much. Thanks for the talk, Ashutosh. I missed a little bit in the beginning, but I think I got just for the rest of it. Okay. So over to you.
Thanks for all the participants who have joined us online. And it was really a very interesting talk. And I think Pradnya has also posted the feedback link, so I would request everyone to kindly give their feedback. I will also send an email officially asking for feedback. So if we've done, I think there's one question which is come. It's from Avinash, he's still there. Pradnya?
Yeah, Vinash is still there.
Okay, so
Last question.
so this is the last question that we'll be addressing. He's asking, every large hospital has a house biochemistry laboratory. How would you convince them to switch to desktop machine for a few drug detection? Please comment on your marketing strategy and competition from Abbott and Roche.
Yeah. So yeah, Abbott or Roche, none of them have anything like this, and we're not talking about $100,000 COBAs that Roche has. We're talking about $1,000 box. So there is no comparison in terms of price. That's one. Two, hospitals and clinics are very different things. This is targeting pain management clinics, which are not hospitals. So which are really doctors. They don't want to even make the changes to become a lab at all, because that's a cost. So they want a clear wave product which their technician can use. So that is the very, very defined use case that we went after.
Yeah.
So to your point, yeah, it's not the hospitals that we were targeting at all.
Okay. Thank you very much.
So I will stop the meeting here.
Thank you very much. Thanks a lot.